Okay, so I am Magnus stand-in. And to illustrate the impact that the microbiota signature can have for gastrointestinal symptoms, our intention is to use irritable bowel syndrome as a proxy for that, because the syndrome in itself includes both all of the symptomatology you may need to illustrate bowel symptoms, regardless if you're talking about constipation, diarrhea, bloating, or whatever is there in IBS. When it comes to trends in research on pathogenesis and pathophysiology in IBS, for a long time gastrointestinal motility was in focus, but during the last 2 decades you can say that the variation in the pathophysiological models have increased quite a lot, and among them the last decade the microflora is more or less exploding in our face. This is what it looked like for now. We have pathophysiological mechanisms of the traditional type, which is related to motor disturbances, visual hypersensitivity, but also we have external disturbances like abnormal central processing of sensations, that we know that not one single entity can explain the full symptomatology, regardless if we're talking about motor aberrations or sensory aberrations. We have problems in just explaining parts of the symptomatology that we see in IBS. And when it comes to the microenvironment, where the microbiota also resides, it's always difficult to understand what are the actual role of the microbiota, the primary and the secondary roles. You have the microbiota can also be disrupted by infections. You have a hormonal system. You have an extremely extensive communication with the neural system. And also, as has been mentioned, depending upon which microbiota you are talking about when it comes to the gut microbiota, the luminal microbiota or the more mucosal, the microbiota residing closely to the mucosa, you might end up with different results. What are the clinical scenarios that have led us forward to believe that the microbiota is of importance in IBS? These are the classical clinical scenarios that I guess most of you here in this room know about. The risk that follows with the gastroenteritis. Perhaps Magnus can be one of our study subjects in the years to come. We'll see. Hopefully not. But there is a risk, there is a substantial risk, almost, this is the strongest risk factor for development of IBS that we know of. We also know that disrupting the microbiota by use of antibiotics, causing a dysbiosis, is related to an increased risk of developing IBS. But we also know that we can use ways of affecting the microbiota by probiotics or non-absorbable antibiotics, and at least short-term have positive effects from that when it comes to the bowel symptoms. These are 2 meta-analyses for the effect of infection of risk of developing an IBS, and you can see they both agree that the increase in risk is 6 to 7 times compared to the unexposed population controls. We also know that it's not just the infection in itself, but also other factors locally in the gut, how the immune system reacts, and also external factors outside of the gut that affects the relative risk of developing long-standing GI symptoms after an acute gastroenteritis. When it comes to antibiotics, this is a UK study, I think, where you compare the occurrence of new gastrointestinal symptoms during a time period, an observed time period, and being treated with antibiotics compared to those not treated by antibiotics, the control population. The risk of developing one new symptom and 2 new symptoms is an independent risk factor for in this situation. The other way around, probiotics, we know that there are very diverging results. Alex Ford will talk much more about this, but there seems to be a class effect that is positive but we don't know which individual species or strains are most beneficial. But the number needed to treat of 7 is not that bad when we're talking about treatment in IBS. And we all know that rifaximin, a non-absorbable antibiotics, has an indication for treatment of non-constipation predominant IBS, not in Europe, but in other parts of the world. And what has been observed is a superiority when it comes to relief of the global IBS symptoms compared to placebo, both short-term and after repeated treatment. So in an integrated conceptual framework on the pathophysiology of IBS, it seems like microbiota might have a central role in most, or can affect or be a part of most of the pathophysiological mechanisms that we have been investigating during the last centuries. Even how the communication between the gut and the brain behaves might be affected by the microbiota. So the interest for our own microbiome is by all means motivated. So are there differences if we compare the IBS population with non-IBS population controls? These are two studies. This first study is Poo from Gothenburg. I would like to disagree with the investigation in athletes. In non-athletes as well, we have easy access to poo. Our patients actually likes to provide us with poo samples. They find it interesting that we are interested in their central problem, the gut. But what we could see here, that this is done in collaboration with the core group, is that if we compared healthy controls or normal non-IBS controls with IBS, we could separate one group with an IBS type signature and one group with a healthy microbiome that had interspersed within it the IBS population as well. This has been repeated by, this is a collaboration in Finland, Netherlands, UK, I think. Let's see where I ended up now. They describe more a kind of a gradient where the amount of IBS patients are enriched in one end of the microbiota spectrum, and the amount of healthy controls are enriched in the other end of the spectrum. And looking at the Swedish cohort, the IBS population could be divided into, say, two major clusters. One cluster that could be separated from the normal controls, and one cluster that was non-differential from the healthy controls. And looking at the clinical features in those two clusters, you can see that the cluster that had microbiota that was similar to the control group had a higher degree of psychiatric comorbidity. This is speculative. We have not been able to repeat it, but you can speculate that in the population with a normal-like microbiota, perhaps the central factors are more dominant. Speculative. And this is a concluding slide made by Mirjana, who will have the next talk on Skype, I think, trying to show all of the studies that has been done and what has been seen in different studies. And as you can see, firmicutes seems to be consistently increased in the IBS population, but when you go down to species, you have diverging results in some of them. You have only in those with bold letters you have this repeated. So there are a lot of studies reporting findings that we have troubles repeating or sometimes are diverging. So what have we done when it comes to the connection between the gut microbial signature and symptoms, bowel symptoms? Well, our latest study is a study that where we have managed to have a signal that certain gut microbial signature can be linked to IBS symptom severity. We started out with an exploratory cohort of 150 patients, and we also had a validation cohort of 46 patients to validate the findings in the exploratory cohort to start out with. We had fecal samples and biopsy samples in the exploratory cohort, and we had fecal samples in the validation cohort. And by a machine learning procedure, now we're into methods that to a clinician is very hard to understand. But with machine learning procedures, we could find 90 OTUs that were enriched in patients with IBS and mostly in those patients with severe IBS compared to healthy controls. If you do this kind of procedure in a random sample, most often you end up with finding having a selection of 50 OTUs by chance. But this one by far exceeds the by chance limit, you can say. And we had the same result regardless if we took samples from the validation stool set or the biopsy set from the exploratory cohort. It seemed like the selected bacterial components, the 90 OTUs, were useful as a marker for IBS severity. And if you look at those 90 selected OTUs, they are spread along the full microbiome. It's not scattered in one part of the microbiome. Each of these dots is one sample. They are scattered among the different bacterial taxa. If you look at the weight that each of these selected OTUs have when it comes to the model that we're using, some of them are associated, those going to red are associated with increasing IBS symptoms. Those going to the green direction are associated with health or less severe IBS. And looking at the healthy controls, the green ones are dominating, which seems to be reasonable. If we combine this finding with the clinical data that we have in these patients, these are cohorts of patients that we have characterized by trying to characterize them from most of the pathophysiological mechanisms that we know of and see how they relate to each other. So if we have one of those PC loading plots, you can say we can have severity here. On this far end, you have the severe IBS population. On this far end, you have those with less severe or the healthy ones. And here you have the scattering of the different clinical parameters, where, for example, IBS-C and IBS-D ends up in the extremes, which makes sense. If we look upon this and look upon what those that have the weight in the model for severity, you can see that those red dots that looks more brown here, I must say, are more far to the left here, which means they are in the severe spectrum of the IBS population. When it comes to the microbial richness, you can say that the microbial richness seems to decrease in the severe spectrum. The size of the different blobs are related to the severity, to the richness, and the color of the blobs are representing degrees of severity or health, where the brown blobs are those with severe IBS. Once again, the scattered to the left. And if you look at methanogens, the exhale in exhaled air, if you can detect methane, you can see that those with the lowest methane in their exhaled air tend to be enriched among those with the more severe IBS symptoms. IBS subtypes does not seem to make a difference at all here. They are scattered randomly. And finally, when it comes to the enterotypes, the major enterotypes, it seems like the blue dot representing Bacteroides are overrepresented in the severe IBS population, or the patient population with severe IBS symptoms. So if you want to conclude this study, you can say that if you have a spectrum ranging from severe IBS to health, the severe IBS patient or the patient with severe IBS symptoms, you can have a severe IBS patients by other means, but those with severe symptoms are characterized by low microbial richness, low methane in exhaled air, which corresponds to undetectable Methanobacterialis, and also that they have a Bacteroides enterotype enriched microbiota. Rather complicated study with interesting results. And this is also something that is gaining interest when it comes to having something in clinical practice. This is a Norwegian company, I think it is, that has this GI map dysbiosis test, where they have developed a scale ranging from low dysbiosis to high dysbiosis, and have been able to show that the high dysbiosis index levels are overrepresented in GI disease, even if it can't separate GI disease, and seems to be less representative for the healthy state. But there's still a lot to learn when it comes to gut microbiota composition. It seems to be altered in IBS patients, where you can by certainty say at least some IBS patients. It seems to be associated with IBS symptom pattern and severity, but definitely a lot of more studies are needed. Thank you for your attention.